contraction of skeletal muscles are initiated by the action potential from the nervous system. The motor neuron carries the action potential impulse from the CNS to effectors until met upon a neuromuscular junction. The neurotransmitter acetylcholine is released into the gap between the axon terminal and sarcolemma of the muscle fiber. The acetylcholine binds to receptors on the sarcolemma. Sarcolemma ion channels open up and make the sodiums move through the membrane. This then generates a muscle action potential, moving along the membrane through the T tubules, causing a release of calcium ions from the sarcoplasmic reticulum to flood into the sarcoplasm.
In order for muscles to contract, the muscle cell's thousands of sarcomeres, aka a unit of muscle contraction, would need to function. Its main components are of myofibrils, with actin, which are thin filaments, and myosin, which are the thick filaments. Z-lines serve as a border for sarcomeres. The H zone is where only myosin is at, and where the supporting proteins create an M line. I band where actin is at only. A band is where myosin and actin overlap. These myofibrils are arranged in this particular way for distinct functions. They slide over each other to contract and relax the muscle, which explains the sliding filament theory. Where calcium and ATP is added, actin myofilaments slide over and myosin myofilaments, but don't shorten. When actin slides over myosin, sarcomere's length is shortened. This, many fibers and sarcomeres in a muscle working together cause movements necessary for organisms.
Run.
Calcium ions released by the sarcoplasmic reticulum bind to troponin, changing in shape, which exposes sites of the myosin heads from the myosin strand used to form a cross bridge. The myosin heads are binded to the actin strand using energy from an ATP molecule. The release of products of the spent ATP molecule, ADP, and an inorganic phosphate signals the myosin head to change shape and bend.
The change in shape of the myosin head and the crossbridge formed between the actin and myosin strands due to the release of the inorganic phosphate is known as the power stroke, which pulls the actin strand towards the center of the sarcomere, causing it to contract. ATP once again binds to the myosin head to unbind them from the actin strand. This process continues until normal action potentials are received by the muscle cells, which causes the levels of calcium ions to drop, which allows the sarcomere to relax.
Wait, how long are we doing this for?
I think we're good. What?
Oh.
It's kind of heavy, Leo. Here.